Hello, my name is Fidon, and in today's video I will present our work on distributionally robust automatic segmentation of white matter, ventricular system, and cerebellum in fetal brain 3D T2-weighted MRI. Deep learning is the current state of the art for this task. If you're not familiar with deep learning, here is how training a deep neural network works. At each iteration, we draw a batch of pairs of 3D MRI and manually segmented segmentation. The network computes an automatic segmentation for each 3D MRI, and the automatic segmentation is compared against the manual segmentation using a loss function. This comparison gives us a gradient that is used to update the parameters of the deep neural network. These steps are repeated until convergence. This equation is the optimization problem that we try to solve using the algorithm that I have just described. It is called empirical risk minimization. When we first experimented with empirical risk minimization, we obtained a very good segmentation performance, at least as measured by the mean and standard deviation of the die score. However, it appeared that the performance was poor on a few cases with abnormal central nervous system. In this work, we study a generalization of empirical risk minimization with the goal of improving the worst-case performance of deep neural network across a variety of fetal central nervous system conditions. We make the assumption that abnormal central nervous system conditions are underrepresented in the training dataset that are available in practice. This is due to the higher anatomical variability in the population of abnormal brains. Deep neural networks trained using such datasets might favor neurotypical cases with suboptimal generalization for abnormal cases. Two consequences. First, we need to change the way we train deep neural networks. Minimizing the mean loss is not enough. Second, we need to change our evaluation methodology. Mean and standard deviation are not enough. Instead, we need a measure of risk, like the percentiles, for example. Now, let's have a look at the math behind this problem. Here is the empirical risk minimization optimization problem that we have seen previously. One can remark that in this optimization problem, the mean training loss is minimized. That is, an equal weight is given to every training case. That is why underrepresented subgroups, such as abnormal fetal brains, might be neglected by the deep neural network. To tackle this limitation, we propose to replace empirical risk minimization by distributionally robust optimization, as defined by the optimization problem on the right. Now, every training case is weighted based on its loss value. A regularization term on the weights is added, because otherwise all the weight would be attributed to the case with the highest loss.
Okay, but how can we optimize this min-max problem?
No problem, the inner maximization problem has a closed-form solution.
And in practice, how does it look like?
If you remember the algorithm for empirical risk minimization, the only thing that you have to change is the sampling strategy. The additional computational cost is negligible, and we have proved the convergence of the algorithm.
Cool, thanks.
In this work, we have also shown that the distributionally robust optimization problem is a surrogate of the minimization of a percentile of the training loss distribution. This result formalizes the relationship between distributionally robust optimization and our initial motivation. Let's now move to the evaluation of the method. The training dataset contains a majority of neurotypical cases, with a minority of cases with spina bifida or other brain abnormalities, which is typical in clinical datasets. For the testing dataset, we managed to gather 98 cases with spina bifida, 54 neurotypical cases, and 41 cases with other central nervous system abnormalities. We have based our experiments on NoNUNet, which is a state-of-the-art deep learning pipeline for volumetric medical image segmentation. We compare the modified NoNUNet, which uses empirical risk minimization, and NoNUNet with distributionally robust optimization. The evaluation is performed using the DICE score, but not only the mean and the standard deviation. In addition, we report several percentiles that are measures of risk. The evaluation is also stratified between controls, spina bifida, and other central nervous system abnormalities. Now, I would like to highlight the improvements in terms of DICE score of more than 0.5 points of percentage in green and of more than 25 points of percentage in yellow. Those improvements all occur in favor of distributionally robust optimization for the abnormal fetal brains and for the percentiles at 10% and 5%. That represent some of the worst cases here. The investigation of the predicted segmentations of empirical risk minimization in the center and distributionally robust optimization on the right shows several abnormal cases for which empirical risk minimization performs poorly as compared to distributionally robust optimization. In particular, there are spina bifida cases for which the cerebral mass was completely empty for empirical risk minimization, but not for distributionally robust optimization. The cerebrum is particularly important for studying spina bifida. In conclusion, we have shown that the high anatomical variability across pathologies can affect the robustness of deep neural networks when they are trained by minimizing the mean training loss. To mitigate this problem, we have proposed to train deep neural networks using distributionally robust optimization, and we have obtained improvements on some of the worst pathological cases. In parallel, we have highlighted the need to rethink our evaluation methodology. First, we need to report measures of risk, such as percentiles, and not only the mean and the standard deviation. And second, there is also a need to stratify the evaluation of deep neural networks, as it was previously discussed in the literature. Last but not least, despite some improvements, distributionally robust optimization does not appear to be sufficient to tackle this difficult problem. And we think that new methodologies are needed to improve the robustness of deep neural networks to anatomical viability in fetal brain MRI. Thank you for your attention.